This is the first lecture for Monday, April 20th. And for this first lecture, I just want to finish up this topic of buoyancy in deep sea sharks, this major adaptation that you find in not just in deep sea sharks, but this is a good example for looking at how different components of the communities in deep water habitats achieve neutral buoyancy. We're just looking at one particular group, but we're seeing that there's a lot of variability. If you looked at fish, if you looked at squid, you looked at crustaceans, you looked at salps, other things that are obtaining neutral buoyancy in the deep sea, you'd find that they employ various mechanisms to attain neutral buoyancy. The selection pressure for being neutrally buoyant is very strong. So it's really, really common that you find neutral buoyancy. Sure, there's things that lay on the bottom that don't worry about being neutrally buoyant. But the point again, you don't have as much energy to swim constantly. You don't have as much energy to have a high metabolic rate that supports that active lifestyle. You've got to conserve what little energy there is. So we were looking at variability within different groups of deep water sharks. We saw that among species, there's the typical way that deep water sharks achieve neutral buoyancy with the big liver, oily liver, these low density lipid classes. But some of the other species that obtain neutral buoyancy do it in very different ways. Not with diacylglycerol ether or squalene. Some of them not even that much with a big liver or an oily liver, but the muscle is an important component. So let's look at the differences with some other aspects, beginning with sex between males and females. For immature sharks, there's not much of a difference between males and females. They don't have the ovaries and testes that have developed. They're very small fraction of their body weight. But in mature sharks, males have a liver oil that contains more squalene compared to females. Squalene is this lowest density lipid class. So even with a little more squalene in your liver and the oil and your whole body is going to be less dense. You're going to be achieving neutral buoyancy using squalene more than other mechanisms. So as a result of that, not surprisingly, because males have more squalene in their oil is less dense, and their liver is less dense. So let's look at these two figures down below to compare males and females. So what the figure on the left with those green arrows shows is that there's three different species that are shown here of the typical deep water sharks: CSQ, CYP, ETP. Those are three different species, and there's males in the blue and females in the red. So each species, CSQ has females and males, CYP has females and males. And for a couple of those shown here, if you look at the amount of squalene in males versus females, CYP and ETP, especially where there's larger sample sizes, the amount of squalene in the liver oil of males is considerably higher than females, at least in CYP. In ETP, males have more squalene in their liver oil than females. Now, the combination of squalene and diacylglycerol ether is similar between males and females in the end. With CYP, males have more squalene and more diacylglycerol ether. But the point is, the oil in the liver of males is less dense, and in fact, the whole liver itself is less dense. So if you look at the density of the liver of males versus females, and remember these are mature males versus females, how dense is their liver? This is their hydrostatic organ. This is what's providing lift for them and making them neutrally buoyant. Well, if you look at the figure on the right, Each one of those ovals shows the liver density of males versus females. So for each species, the males in that oval are less than the females. The liver density of the males is quite a bit less than that of the females. Liver density of the males is less than females. If you look at every one of those species, the males have a lower density liver. Their liver provides more lift. And why is that? Because they've got more squalene, they've got more diacylglycerol ether in some cases. Their liver is what they're using for buoyancy. Well, what are females doing about this? Why is there a difference between mature males and mature females? Well, the explanation lies in the fact that these mature females have eggs, and these eggs contain yolk, and that yolk contains lipid, and that lipid contains these low-density lipids. So the females are swimming around. There's a female that's cut open. You can see these big eggs. They're pretty big. They're as big as a little billiard ball in some cases, in sharks that aren't that big. So these females are swimming around with these little eggs that are like little balloons in a way, that are providing buoyancy because they're filled with oil. They're pretty oily, and they contain these low-density lipids. If you look at the figure on the right with these different species, there's different species, and what these figures show is the lipid classes that are found in the yolk of these females that are carrying around these eggs. And so the yellow is squalene. So you see this one species has a huge amount of squalene, and then you have these other ones that have less and less squalene, but still 30%, 20% of the oil in their yolk is squalene. And then the magenta color there is triglyceride, which is a low-density lipid, not compared to squalene and diacylglycerol ether, but it provides a certain amount of lift compared to heavy tissue. And then that light purple is diacylglycerol ether. So you see, for some of these, 50% of the oil in their yolk is a combination of squalene and diacylglycerol ether for some of those species. So they're carrying around these little packets of buoyancy in their ovaries. And it's no wonder that males and females have different densities, because the females are deriving all this lift from their eggs. They don't need to use their liver in the same way that males do. Males don't have those eggs with all that oil, with all that low-density lipid, and so they have to have another mechanism for providing neutral buoyancy. And for the males, that is their liver, squalene, oil. Well, another difference that you find amongst these different deep water sharks is the size of the sharks makes a difference. So if you look at the small sharks versus large sharks, if you look at the percentage of squalene, the lowest of all density lipid classes, the one that provides the most lift, there's more squalene in small sharks, relatively more squalene in smaller sharks. Squalene is a higher percentage of their liver oil. As you get bigger, the amount of squalene, or the proportion of squalene in their liver oil, decreases. So these small sharks have livers that are more buoyant. And that's shown in this figure on the right, where in this case, density, the lower the density, these are in negative numbers, those parentheses. So 10 is more dense than 12, which is more dense than 14. The lowest density is 20. That's really low density. And that's how dense their liver is. So you see this line that's fit to the data shows that bigger sharks have livers that are more dense. Smaller sharks have livers that are less dense. And that's the trend in these species that have been examined, where there's a big enough sample size to look at a range of sizes. So what was going on in terms of difference in these sizes? Well, the small sharks might have more squalene, proportionally more squalene. However, in the big sharks, they have a bigger liver and they have more oil. So the small sharks might have more squalene, but their liver is proportionately smaller. The amount of oil in their liver is proportionately less. So big sharks, as they get bigger, their liver increases in size. And as they get bigger, the proportion of oil in their liver increases in size. Those are two changes. And then, not unexpectedly, since the amount of squalene and diacylglycerol ether are inversely related in a lot of these species, bigger sharks have more diacylglycerol ether. So the squalene is decreasing in proportion as the sharks get bigger, but the proportion of diacylglycerol ether is increasing. Now, diacylglycerol ether has a density of 0.89 and squalene has a density of 0.86, so it's not an even substitution, but still it contributes to a certain amount of buoyancy and low density. And if you look at how dense the body of these sharks is, or if you look at whether they're neutrally buoyant or not, then it doesn't matter what size they are. When these sharks are very small, they tend to be somewhat dense, so they've got to make some changes pretty quickly within the first few months of their lives to start attaining neutral buoyancy. They're not going to be able to survive it that long. But throughout the whole range of these species, they're all neutrally buoyant, which is what you could say about deep sea sharks in general. All these different species are neutrally buoyant. Males, neutrally buoyant. Females, neutrally buoyant. Big sharks, neutrally buoyant. Little sharks, neutrally buoyant. So some of the things that we looked at, this variability amongst species, if you look at this figure on the left where it says the plunket shark, and Aprosteris, you could throw the six-gill shark in there as well. Well, what these are doing to achieve neutral buoyancy, they're all neutrally buoyant, but the plunket shark has the biggest liver and some of the most oil in that liver. The one in the middle, the average squalid shark, now that's the typical deep water shark where they've got a relatively big liver, relatively large amount of oil, and relatively large concentrations of squalene and diacylglycerol ether. So that's the typical in the middle. And then there's the Aprosteris, which has not very much squalene. One species did, but the other ones didn't. Not a very big liver, not that much oil in their liver. But the blue there is this very watery muscle. The result is that the plunket shark and the average squalid shark, Aprosteris, are all neutrally buoyant. If you threw in the six-gill shark, then the six-gill shark would have very little squalene. So the red would be just a tiny little trace, if anything. But similar to the plunket shark, it would have a very big yellow component where the liver is very big and the oil is very big. But what's not shown on this particular figure is the percentage of lipid or muscle or fat, oil in the muscle. And that would be 10 times higher in the six-gill shark. The result is everybody's neutrally buoyant regardless of the species. If you look at big sharks and small sharks have more squalene, but bigger sharks have a bigger liver and more oil, neutrally buoyant. If you look at males versus females, males have more squalene, but if you look at their body outside of the liver, then females are especially low density. So it's similar in a way to what A. Pristurus and the big deepwater sharks are doing in using muscle for additional buoyancy outside of their liver. But females are using their eggs and ovaries. In the end, though, everybody is neutrally buoyant. Strong selection pressure for neutral buoyancy. But there's different ways that you can attain neutral buoyancy in this relatively small group of animals, deep sea sharks. Liver size. You can have a really big liver and then sacrifice some of these other mechanisms of increased buoyancy. You can have a lot of oil in your liver. That number 2, liver lipid content, that can be really high, and you can sacrifice some of these other mechanisms of buoyancy. Liver lipid composition. You can have a lot of squalene. You can have a lot of squalene and a lot of diacylglycerol ether, or you can have diacylglycerol ether. There's a lot of variability in the composition of liver oil. So you can use that, and that's what the typical deepwater shark does, is use squalene. Another difference that is quite extraordinary in the big deepwater sharks that probably have a limit to how big their liver can be is the lipid content of their muscle. 6-gill sharks, sleeper sharks, these big deepwater sharks have oil in their muscle, provides a lot of buoyancy. There's also variability in the amount of water in their muscle. If you have a watery muscle, you can get by with having a smaller, less oily, lower composition of low density lipids in your liver, and that's what cat sharks do. You can have lipids in your eggs, not just any lipid, but you can have these low density lipids, diacylglycerol ether and squalene. That's what females do. So manipulating all these 6 different characteristics, in the end, everybody's neutrally buoyant, but there's differences among the species, there's differences between males and females, and there are differences in the different size classes. So that's quite a bit of detail about buoyancy in deepwater sharks, but it shows you the different selection pressure and the different adaptations, different evolutionary histories of different species, all trying to solve the same problem of achieving neutral buoyancy and being able to benefit from those characteristics of being able to float, basically, save yourself a lot of energy. But they go about it very different ways, very different mechanisms for answering that same question.